everyone, and thank you for joining us for this evening's live stream presentation. This evening's presentation title is It's the Little Things in Life, Characterizing Wildlife Microbes with Andrew Bartlow and Kane Moser from the Biosecurity and Public Health Group at Los Alamos National Laboratory. My name is Kristen O'Hara. Hi, I'm the Director of Interpretation at Baja Mental Education Center, or PEAK, as we like to call it, which is located in Los Alamos, New Mexico. I will be the moderator for tonight's talk. So as many of you know, PEAK is the nonprofit that operates the Los Alamos Nature Center. Please visit our website, peaknature.org, that's peecnature.org, to learn more about our organization and our upcoming events. I would like to give a quick shout out to all our wonderful members, donors, volunteers, and staff. We really wouldn't be able to make any of this possible without you. So thank you so much. To learn more about becoming a PEAK member or donor, please visit peecnature.org. We are joined this evening by Andrew Bartlow. They are staff scientists at the Biosecurity and Public Health Group at Los Alamos National Laboratory. He is the co-leader of the Ecological Health Security Lab, where they combine research, training, and international science networks to tackle local and global health security challenges driven by changing ecologies. Their research projects focus on ecology, transboundary transmission, and biosurveillance of emerging zoonoses and neglected diseases. They take a One Health approach to reduce disease risk. Love that. Kane Moser is a post-bachelor's student in the Biosecurity and Public Health Group at Los Alamos National Laboratory. In addition to microbiome research, they also studied the West Nile virus transmission cycle with a focus on the biology of mosquito vectors and avian reservoir populations. I thank you both for joining us this evening and sharing your knowledge with us. And I'm going to go ahead and turn it over to both of you. I believe Andrew starting, and I will monitor the chat.
All right, great. Thank you. Thanks for the introduction. So I'm going to get started today. I'm Andrew. I'm going to turn off my video because to save some internet. But yeah, I'm going to share my screen. Hopefully everybody can see that. Does that look good?
Yes, it looks great.
Okay, great. All right, so yes, my name is Andrew. I'm going to be co-presenting this with Kane, a student in B10 Biosecurity and Public Health at Lano. And we're going to talk a little bit about characterizing wildlife microbiomes and why they are so important. So I'm going to start off by talking about biological communities in general. And so what biological communities are, they're just a collection of species in a given area. And the difference between a community and a population is that a community is all the different species within a given area, and a population is just one species. And so you can talk about a population of bluebirds or a whole community of birds here on the Pajarito Plateau. And you can talk about communities in terms of mammal communities or large vertebrate communities in Africa, or desert animals of Death Valley, community of amphibians, etc. And so it's no different that when we're talking about biological communities, microbiomes are just communities of bacterial species in a given area or in somebody's gut. So I'm going to start by talking about sampling biological communities and what they have in common. And so you could go out and survey insects in forest patches. You can take your little net and you can go and sample anything that you find and identify it to species. You could go out and make quadrats and survey vegetation in certain areas. Or you could go out and trap small mammals in a forest in Pennsylvania. And all of these things are going to give you the same type of data. That's the species and the relative abundance in a given area, and that gives you community data. And so you can get things like, and I'll explain this in more detail in a little bit, but things like species richness, which is just the number of species, species abundance, the abundance and how much of those species are in a given area, species diversity, which kind of is an index, taking those two measures into account, and also species composition, like what is it? What are the species there? Is it a bluebird and a flycatcher, or is it some other group of species? And so I am going to, yeah, and so can we use community data as signatures of environmental change? And I'll bring that up as we go along. But I wanted to start off by talking about what's happening in our own backyards. And I know you're probably thinking that we should talk about like microbiomes, but I think this illustrates a good point about biological communities and responses to environmental change. And so back in 2002 and 2003, there was a big drought in Los Alamos and on the Pajarito Plateau and other places in the Southwest. And after the drought, the trees were stressed and bark beetles came in and just wreaked havoc on the pinyon pine trees in the area. And in 2003, about 24 to 97% of the pinyon pine trees were dead. Just 1 year later, over 90% of the pine trees were dead. And by the end of 2013, all of the sites that we were looking at had greater than 98% pinyon mortality. So that is a pretty big environmental change, right? In 2004, there was, in some places 95% pinyon pine tree mortality. So no more of these big trees that birds like. And so we wanted to know, how will these birds respond? And so we went out and my advisor, Jean Fair, some of you may know her, did point counts in the area and found that, from 2003 to 2013, and we found that there was a big decrease in the total number of species and the total bird abundance and also species diversity. And so this is pretty drastic. So in terms of like total abundance, right, almost 250 species were found in the area in 2003. Pinyon mortality happened, and then we see a big decrease in the abundance of birds. And so now we're only getting about, in 2013, about 50. So we really wanted to know what is happening. Are these birds dying? Are they disappearing? Are they moving up in slope? And we looked at bluebirds specifically, bluebird population, one species, and we found that from 1997 until 2015, these birds were nesting. So this is annual nesting elevation from all the way down to White Rock, all the way up until Los Alamos. The average nesting increased over time. And so they are increasing their elevation to cope with some of these pine tree deaths. The problem is, and so here's some of the species that were lost in some of these lower elevation sites with a lot of PJ mortality, their pinyon pine mortality. So this is a really big problem. We don't want to wait 10 years for something like this to be evident. And so this is because of this process called extinction debt, which is just that things take a long time to disappear or go locally extinct when there's some type of environmental change. So we don't want to wait years and decades for these things for us to realize that something's happening. So we need to identify the effects of environmental change much more effectively. And instead of multiple decades, we need to do it in maybe a few months or a year. And so I just talked about maybe using biological communities as signatures, but looking at actual wildlife populations and wildlife communities, that's going to take a long time. So we thought that we could start looking at microbiomes of individual wild animals because the same concepts apply. It's a community of bacterial species, and you get an idea of abundance of those species, how many species there are, and because it's all the same community data and all the same concepts and statistics and everything apply. So you can analyze it the same way. And so this is just microbiome research has exploded within the past decade or two, and people are still finding ways about, how the microbiome influences an individual's health and in wildlife ecology and evolution, which Kane will talk about here shortly. But typically, right, you have a healthy microbiome with a lot of these friendly microorganisms, a lot of diversity, a lot of different species doing a lot of things and to help you out. And that's gonna, produce, make you healthy. On the other hand, you can have an unhealthy microbiome with a lot of these unfriendly microorganisms, maybe some pathogens get in there, and they are, maybe not so healthy. And so they've done some studies in mice where they've taken the microbiome of healthy mice and transplanted it, done a fecal transplant into obese mice, and it actually helped the obese mice. And so there's this idea that microbiome is really important for the health of an individual. And so, yeah, things like decrease in species richness or abundance, or difference in composition, or pathogenic microbes are more common. Maybe these things could point to environmental change and wildlife populations and communities being stressed. And so the idea is to take things like I talked about tree mortality, since all of these things influence wildlife communities and populations, but this takes multiple years and decades. So our ultimate goal is to look at the microbiomes of these individual hosts. We can still get the same data, but over much shorter timescales. And so in order to do this, there's a lot of variation in wildlife microbiomes among even within an individual species. And so things like age, male or female, health status could impact what species and what the diversity of a microbiome actually looks like. So we really need to understand the natural variation in these bacterial microbiome communities. And so, yeah, the ultimate goal is can we predict environmental threats much faster? And this is really important for things like amphibians, and you can get an idea of how well protected certain species are from chytrid fungus, for example, and maybe bats with white nose. So understanding these things is hopefully we can predict these things faster and aid in conservation. So our lab has a lot of microbiome data sets. We have some on California condors, which are recently reintroduced. We're going to talk today about the screech owl microbiomes, two species, one that is pretty, the Western screech owl is pretty abundant in the West. And then there's the whiskered screech owl, which just gets barely into the United States in Southern Arizona and Southern New Mexico. So we're going to talk about samples that were collected down there. But I'm going to turn it over now to Kane, and they are going to talk about a little bit more in detail about microbiomes. I'm going to stop sharing quick.
Hi, thank you. So again, I'm Kane, and I'm going to give a little bit more background on the history of research on microbiomes, and then some of our work as well. And I'm going to turn my video off too. Okay, can everybody see that?
Yes, it looks great.
Awesome. So yeah, so just to start with some more basics. So what is a microbiome actually? So as Andrew already mentioned, it's just a community of microorganisms living in or on an animal host. And we've actually been interested in microbes really ever since Van Leeuwenhoek first observed what he called animalcules, which honestly, I think we should still call them that. And then if we fast forward about 200 years, the next major milestone came with a book by Joseph Leidy, considered by some to be the origin of modern research on microbiota, called Flora and Fauna Within Living Animals. And then for the next 100 years after that, the research community was really focused on germ theory and eradicating microbes that cause disease. And so then in the 1960s, a group of researchers discovered that germ-free laboratory mice were actually lacking aspects of normal physiology. And so that kind of kicked off this idea that, oh, maybe not all microbes are bad. Maybe there's something to this. And since then, most research on quote unquote good microbes has focused on human health. So way before Van Leeuwenhoek ever saw the animalcules in his microscope, there in China in the 4th century, there was actually a man named Ge Hong who published a Chinese Handbook of Emergency Medicine. And this was the first kind of recorded example of using fecal microbiota transplant to treat some sort of illness. And that's pretty interesting. It was a very long time ago. And then in more modern times, FMTs were first applied and seen to be successful for treating an illness, pseudomembranous colitis, in the late 1950s. And again, as I already said, most research since then has really centered human gut health in particular. So the field of microbiome science has really exploded in the past couple of decades, but it is still in its infancy. There's so much more to be done. So why do wildlife microbiomes matter? Well, the microbiome in general plays a really essential role in animal host physiology, behavior, and health. And in terms of the human microbiome, there's a lot known, but there's also a lot that's still unknown. So in terms of diversity, and then also even more so in terms of functionality. And for wildlife, these numbers are even higher, really high. There's not much known about wildlife microbiomes at all. And so in this next section, we're going to cover a little bit of ecology and evolution, abundance and distribution, disease risk, and the effects of climate change in the context of wildlife microbiomes and why they're important. So in mammals, gut microbiome colonization has been shown to influence nutrition, brain development, and behavior, especially stress response. So it pretty much influences every major aspect of biology. And animals have evolved to work with the microbes that live in and on them to actually improve their evolutionary fitness. And within this, diversity matters. So the more, as Andrew mentioned, the more, excuse me, the more the higher number of species present in a microbial community within a host, often the more the impact on increased fitness is, but that is only one half of the equation, or one part of the equation, because community composition also matters a lot. And a lot of animal host species are really highly specified in terms of their community composition of their microbiomes. And microbiomes are so important to host health that they're sometimes referred to as another organ. So just to recap, I think Andrew mentioned this already, but there are two types of diversity that we look at in ecology typically. Alpha diversity is just the number of different species present in one community. And beta diversity is a way of examining similarity or dissimilarity between two different communities, or more than two different communities. And composition, on the other hand, is more about the identity of what is in those communities, who is there, and the relative abundance of those species in the community. So as I mentioned, microbial community composition is host specific. And in terms of distribution, or abundance and distribution rather, the microbiome affects new ecological capabilities. So an example of this that could be maybe applied to abundance is that amphibian skin microbiomes actually protect them from their environment. And so, if you have protection from your environment, you can probably do a bit better at reproducing, and there will be more of you. And as an example for distribution, vulture gut community composition has been shown to actually have enabled them to inhabit new dietary niches by eating carrion. And that's due to the specificity of their gut microbiome and actually brain, I think, I forget what it is, but something to do with their heads. the composition of their microbiomes. So what species of bacteria are there? And so again, this can allow host animals to expand their ranges and affect their distributions. And then in terms of disease risk, the microbiome's impact on an animal or host animal's ability to transmit disease or its likelihood of getting sick. Dysbiosis is a really important concept. So a healthy immune system is in a state of symbiosis with the microbiome. And I'm sure everyone's heard the term symbiosis, but it just means that, two things are working together, basically. And the microbiome plays a really key role in immune function. And so predictably, dysbiosis is just an imbalance or disruption of the normal microbial communities. And this increases disease risk. So when your normal microbiome is disrupted, your immune system is not functioning as well, and this increases your susceptibility to pathogens. And an example, a really sad example of this is the chytrid fungus that affects amphibians. It has wiped out a ton of different species and has been really devastating for a lot of amphibian communities. And this is clearly linked to their microbiome and changes occurring that make them more susceptible to chytrid. And so what causes dysbiosis to occur? Well, a lot of different things, but all of them are pretty much tied to external factors. So changes in the habitat of the host, broader kind of environmental changes, stress on the host, etc. And how we are able to observe this and know these things is by looking at altered composition and diversity and the cascading effects of dysbiosis. So once there's an imbalance present in the microbiome, then that affects things, again, like the immune system, stress response, and then that cascades into other elements of the host biology and ecology, ultimately negatively affecting their ability to survive. And so how is climate change linked to this? Well, host-associated microbiota are affected by anthropogenic activities. There's a lot of evidence for this. And some of those activities include habitat fragmentation, heavy metal exposure, and especially there's been a lot of research on captivity. The microbiomes of captive animals are really, really different from wild animals. And across the board, they are less diverse, and those animals are more susceptible to pathogens, et cetera, and more stressed. So climate change impacts every aspect of animal biology. In particular, increasing temperatures are like one of the most major factors. So the negative effect on host animals of climate change or of increasing temperatures is actually more closely linked to changes in the microbiome than to direct thermal stress. A really cool study came out on this, showing this. So in other words, the way that heat, like high heat, is impacting the microbiome, the cascading effect sort of that on the host animal is actually like more negative effect on the host animal than just the host animal being exposed to high temperatures and whatever effects come from that, if that makes sense. So climate change really matters to microbes, and obviously microbes really matter to the host animals. So as Andrew also mentioned earlier, the effects of climate change on microbiome composition and diversity could actually shape the responses of animal host species to the same shifts in climatic factors. So now I'm going to shift to talking about some methodologies, some tools, and some techniques that we use in the field. Or sorry, yeah, I guess. Yeah, so just kind of an interesting thing is that there is currently no standardized best practice for microbiome sampling in wildlife. So there's no how in the lab there are very specific protocols that you need to follow to make sure that you are basically doing things based on precedent and doing experiments in a way that makes sense. But because this field is so new, there's not a ton of precedent. So when you're out in the field collecting samples, there are kind of two main types of microbiomes that are typically sampled. The first one is the gut microbiome, which is the most heavily studied. And there are three different ways of obtaining those samples. The first one is destructive. It kills the host animal, and that is by actually taking the intestines. And the other two are more commonly used now in wildlife especially. And those are fecal collection and rectal swabbing. And then the other type of microbiome that is often studied, especially in amphibians, is the skin microbiome. And those samples are obtained by swabbing, skin taping, which is kind of weird to think about, just like applying a piece of tape to like a frog's skin. I don't know that much about it, but it seems funny. And then fur and feather collection as well. And so then moving on to how we get those samples into a form that we can kind of analyze and understand, takes us to next generation sequencing. So sequencing is basically any process that allows you to determine the order of the nucleotides that make up DNA and RNA. And the first protein that was ever sequenced was actually insulin, and that was done by Frederick Sanger in the 1950s. And that is where the name Sanger sequencing comes from. And so next generation sequencing utilizes high throughput technology. And what that means is, so it's actually a concept, I believe, from computer programming. It's massively parallel processing, which basically just means that you can perform many different computations or tests, et cetera, in this case, sequencing multiple DNA fragments at once, all at the same time, so concurrently. And so this allows large-scale sequencing of whole genomes and targeted genes, and then even targeted areas within a targeted gene, as I'll talk about. And so Sanger sequencing is still used today, but it's not as like massively scalable. It's mostly used today for kind of smaller projects, or not smaller projects, but projects with smaller sample sizes.
All right, you can just keep your slides shared and I can tell you to go. Yeah. So sequencing. So yeah, like Kane mentioned, we actually, to get microbiome data, to get samples, we actually have to go out in the field, collect the animal, whether it's a bird using a mist net or a wildebeest using tranquilizers in Kenya. You still have to get that sample, you have to swab that animal, you have to get that sample. And because we're ultimately interested in the microbiome, we need to do the sequencing, which is highly dependent on contamination. So even though we're going out in the field and trapping these animals, handling them, sampling them, we have to use the best practices to prevent contamination, either from the environment. You don't want to drop your swab that in the soil, in the dirt that you are planning on sampling, or maybe you just swabbed. And a lot of contamination is you as a human. And so we have to make sure that we're wearing gloves, sometimes double gloves, and making sure that we're using all sterile equipment and things like that. So ultimately, yeah, we're going to bring the samples back to the lab. Another thing that is really hard to do is keep samples cold. As some of you are aware, DNA and RNA aren't very stable at room temperature, and they degrade very quickly. And so we need to keep the cold chain going, and whether that's from taking a sample, putting it in a cooler with ice packs, getting it in liquid nitrogen or dry ice, and then ultimately in a -80 freezer. Sometimes that process is hard and hard to figure out how that's going to work. And in the lab, it's great. It's easy to do it on mice, but once you get into the wildlife area, this becomes a big challenge. And so ultimately, we want to go into the lab with really good samples, really high quality data, no contamination, good DNA and RNA, and we want to do sequencing. And so on the upper right is just one of an Illumina sequencer. It's called a NextSeq. We have one at Lano. And what you can do is, all the microbes in your sample are there on the DNA is present or RNA. And you can do a few different types of things to sequence the microbiome data. You can do Amplicon sequencing, which is just sequencing one gene or a couple genes that are kind of good marker genes or good barcoding genes that once sequenced, you can say like, oh, this is an E. coli, or this is another pathogen, or this is something else. And so you can get all of these. So for bacteria, it's 16S is the gene we sequence, and you can get just a library of all your bacterial species that way, since these are they're very good at identifying species. The other hand, you can do metagenomics or metatranscriptomics. So we call, when we're sequencing all the DNA in a host, no matter if it's a bacteria or DNA virus or fungi or any of the food they ate, that's called metagenomics. We're sequencing everything. Metatranscriptomics is specific to RNA, and that's more of it's really important for like RNA viruses, for example. And so next slide, please. You can also do metabolomics, which is just looking at some of the metabolites that are produced from these species. Next. Yeah, so you can do, there's all these sequencing technologies. There's short reads, which are sequencing just a few reads or a few base pairs, a few nucleotides at a time. So just kind of these short fragments. Or you can do long reads. So there's this Oxford nanopore, it's called the Min ion, and it's really small. It fits in the palm of your hand. And this is cool because you can take it out in the field, do a little DNA extraction and a little library prep, and you can stick it right in the sequencer there and get your data, which is super cool. And so, yeah, so you can do, you have how all your microbiome genes here on the different colors. If you do shotgun sequencing, you're sequencing everything in that sample, including, like I said, fungi, bacteria, any other eukaryotes that are there. Or you could go for the phylogenetic marker gene, like a 16S gene, and that's going to be much more limited in terms of data and what you can say about your sample. For 16S, for example, you can only get bacteria. For shotgun sequencing, you can get bacteria, viruses, fungi. So there are pros and cons to each. So we'll mainly be talking about some of the 16S data today, but I think it's really cool. So next slide, please. Of this idea of metagenomics, because it's a really cool way to get at what organisms are out there. And so this idea of shotgun metagenomics is sequencing all the DNA or RNA present in the sample. It's called shotgun because there's no targeted gene amplification, like in the 16S, for example. And we need to use tools to determine what organisms are actually represented by those sequences. And so we have big databases. Actually, GenBank, which is a big one, was actually started at LANL. On like pieces of paper. So like this sequence matches to this species. So we have these huge databases that we're trying to match sequences to known species. So you can do this with a soil sample, environmental sample, lake pond samples. You can do this with sampling livestock or wildlife or humans, air. We have a project going on sampling air, looking at filtered air. So that's kind of cool. So this is a really cool technology, but it is costly and time consuming. So that's kind of a downside to this. Next slide, please. Yeah, so traditional microbe. So this is important because traditional microbe identification required culturing isolates. And so you would grow your sample on a plate and some bacteria would grow, and you would pick that culture and sequence it and get an idea of you could sequence and use that, but most of the time you would say, oh, this bacteria doesn't grow on this kind of plate, it doesn't metabolize sugar, or it does, or it's an anaerobe, or it's gram negative. And so this was not a really good way to do that, and only a small portion of microbes are actually culturable, and so that is also a limitation. So this type of sequencing does not require any culturing. And so it gives a much more unbiased view of everything that's out there, everything that's in your sample. Next slide. And so microbiome data looks like similar to like bird data in a forest. So think of the sites here as like forest 1, forest 2, forest 3. And then we have a bunch of bird species, species A, B, C, D, E, and F. And we just have the number of birds that were counted in those forests. For microbiome sequencing, each sequence that we get to understand the nucleotides of all the A's, G's, C's, and T's. Each one of those fragments is called a read, a sequencing read. And so our reads, our sequencing reads, will be used for the relative abundance. And so we can calculate up all those reads that belong to a given species, and we can use that as our relative abundance measure. Next slide. So this is what some typical microbiome data looks like. We have our species up top, and each row in the table is a site or a sample. And all the numbers within each cell are just the number of reads. So this produces massive amounts of data, and typical shotgun data set will be several gigabytes, and you open up a file and it's basically AGTC, all, millions of them. So we need programs to be able to go in and start to match up which reads look like certain species. Next slide. So certain things, I'll just go through this quickly. So there are some ways to compare microbiomes. You can look at microbial phyla. So if you remember from high school biology, the order of the classification system, kingdom, phylum, class, order, family, genus, species, you can look at really broad phyla. And so there are some results where decrease in certain bacterial phyla, such as this bacterium here, was documented in the case of Crohn's disease patients. And so it's kind of another, what Kane was talking about, that the microbiome really could influence your health and what disease you have or susceptibility to certain pathogens. Next slide. So again, species richness and just the number of species, and the diversity is the relative abundance and number of those species. So I'm not going to dwell on this more. Next slide. Rarefaction is kind of a cool term, and it's just based on this idea of species accumulation. And so on the bottom, on the x-axis, we have number of reeds sampled, which that could also be number of forests sampled or number of individual animals sampled. And so on the y-axis is the number of species. And so as you increase the number of sites or individuals that you sample, you're going to increase the number of species that you find. So you go out to one forest, you sample 10 birds, maybe all 10 of them are different species. And the idea is to see, all right, so we sampled maybe 40 sites, and we get to a point where we sample and sample and sample, and we're not getting any new species. This, as you reach an asymptote, like in let's see, B3, that one's pretty flat. And so you could sequence 1 million more reads here or 1 million more sites, and you're not going to get any more species because you got them all. And so this is an idea of coverage and how much you can actually get in your sample. Did you sequence or sample thoroughly enough? Next slide. The last one of this is a way to look at, it's called Nmds. It's a way to look at community composition. And so these cool plots just take a really big complex data set like species in different areas and distills it down to 2 dimensions. And so in this plot here on the left, and points that are closer together are more similar than points farther apart. And so you can start looking at, all right, this one all the way on the left is very different from this green one all the way on the right. And this is based on the type of species, the names of the bacterial species that are there. And so, yeah, it gets an idea of how similar are these communities. And we would predict that, maybe certain species or siblings in a nest would have similar, the points would group closer together than maybe another family. So next slide. And then there's this last thing that's kind of gaining traction a lot more recently is this idea of core microbiome. And it's what are those species that are shared among most samples or most hosts that are sequenced. And so, yeah, so there for this example, an OTU stands for Operational Taxonomic Unit, which is basically a species. And so you can get an idea of, all right, out of all my individuals, what are the bacterial species that are shared? And so it can get an idea of those common species and maybe things to be as a signature of something happening. If you don't see those really important species, then that could point to something happening or some type of environmental change. Next slide. And I think you're up, Kane, again.
Yep. So now I'm going to talk about a little bit of our research on comparing these screech owl microbiomes that Andrew mentioned earlier. So why did we want to do this? Oops. Well, as I mentioned, wildlife microbiomes are really understudied. There's a lot that remains to be found out about how they work and the composition, diversity, specificity due to the host, et cetera. And we really need a baseline understanding of all of this variation for different taxa, different types of animals. And so the Western screech owl and the whiskered screech owl share an ecological niche. And that really means, as you can see on this map over here, the green is the Western screech owl, the purple is the whiskered screech owl. They share the same habitat, the same types of prey, and even the same nesting preferences. So the same types of nest boxes. And so we really wanted to find out, because these species have such similar ecologies, do they also have similar microbiomes? So the goal of this project was really to set a foundation here and help understand the natural variation present in wildlife microbiomes. So these are just some fun pictures of owls from our sampling in southeastern Arizona. And so the panels A through C are the whiskered screech owl, and the last one is the Western screech owl. They are super cute. And so we basically, I can go over this really quickly, but we had all of these nest boxes, checked them, and then banded and sampled the nestlings using a couple of the methods that I talked about earlier, the fecal and cloacal sampling, the non-destructive ones. And then in order to get this data in a format that we could understand and analyze, we did something called targeted 16S sequencing, with which Andrew talked about a little bit, but basically so actually I just wanted to say the 16S gene is really interesting because it's highly conserved in all forms of life that are based on DNA. And so it's a really useful target for kind of classifying microbiomes from samples that are super complex. So like if there's a lot, like thousands of different species in a sample, how do you parse them out? And so 16S sequencing is really, that's kind of what it does. And so the standard 16S method really targets just one region of the gene, and it relies on OTU binning. And Andrew said what OTU stands for, Operational Taxonomic Unit. And binning is basically just grouping the reads from each fragment into clusters to identify the organism. And our approach was slightly different. So we were able to target 2 regions of the 16S gene, which can provide higher taxonomic resolution than OTU binning. And this method also included a bioinformatics pipeline. And our work was the first application of this new 16S method to wildlife microbiomes. So another goal of our project was really to kind of test it out and see if it worked really well. And it does, it's really cool. And then for kind of actually understanding this data, so making sense of it, what are the different patterns? We had these 2 different sample types, cloacal and fecal, for these 2 different species of screech owls, the Western and the whiskered screech owl. And we wanted to understand species diversity, and this is the same picture that Andrew showed earlier. And on the left is richness, which is like the observed diversity, basically. It's just how many species are there, basically. And then Shannon diversity, which is another measure of diversity that results from a statistical test. And then the other thing we wanted to investigate was community composition. And we achieved this with NMDS analysis, the non-metric multidimensional scaling, I think. And also by looking at the core microbiome, both of which Andrew explained a few slides ago. And so that takes us to our results. So in terms of diversity, just to talk about these plots a little bit. So in the first panel, we're just looking at the cloacal samples. And the first graph is the observed diversity or the richness for both species, Western and whiskered. And then the second is the Shannon diversity. And that's the same in panel B, but just for the fecal samples. And so what we find here is that diversity is actually pretty similar between species and sample type, the diversity of the microbiome. However, in the cloacal samples, species richness was higher in Western screech owls than in whiskered. And these are box plots, by the way, which are just like really cool way of representing like the mean and kind of the bounds of your data. Yeah, so, and then in terms of community species composition, these are those NMDS ordination plots, so spatial grouping of samples based on similarity or dissimilarity. And what we really find here is that owl species and nest box were correlated with microbiome composition. So diversity was kind of similar. However, composition is not. Well, actually, that's not true. Composition is just like a really important factor. So it's different between different species, and it's also different between different nest boxes. And so like one species, for instance, the Western versus the whiskered, those points are going to be grouped closer together because their microbiomes, like the identity of the species in their microbiomes, are more similar to each other than to the other species. And same with nest box. So the same nestlings in one nest box are similar to each other and different from the nestlings in a different nest box.
Yeah, and those numbers by the points are the nest boxes where the nestlings came from.
Yes, thank you. And then for the core microbiome, the first panel is, or the first figure is the Western screech owl, the second is the whiskered screech owl, and the third is the combination of both. And so the core microbiome is really just this idea of like what species are most prevalent here and which ones are shared between these different groups that we're looking at. And so for both species, there was one organism that was the most common. And Andrew, I don't know if you can, I don't really know much about what that is or how to pronounce it. I can try. Blautia hydrogenotrophica. There you go. But the kind of the takeaway, one of the takeaways here is that for the whiskered screech owl, there were twice as many bacterial species shared among nestlings compared to the Western screech owl.
Yeah, that bacterial species that's shared is really important for degrading plant material, which may point to the owls consuming like herbivorous arthropods or some type of plant material. Or it's found a lot in chickens actually, but there's very few reports of it in wildlife and in wild birds. So that's kind of cool.
Okay, cool. So it's probably tied to like their ability to digest things that are common in their diet.
Yeah.
Okay, so these results basically show us that owl species and their immediate environment both influence the composition of their microbiome. So siblings are similar to each other. And this could potentially be due to them being fed really similar things, because when you think about it, all the little baby owls in the one nest box are all being fed by the same parent. And so they're probably eating really similar things. And so this could be influencing this pattern in their microbiome composition. And another interesting thing about these results is that there are differences in microbiome variation between nestlings of different species. So the range of the data, basically. So there's like greater variation. And pretty much across the board, there is greater variation present in the Western screech owl as opposed to the whiskered. And so there was greater variation in the cloacal and fecal samples for observed species richness, and also in Shannon diversity just for the fecal samples. And also a greater dispersion in species composition. So if we think back to those ordination plots, there's a greater, like, the points are more all over the place, basically. And so we're not really sure why this pattern exists, but it should definitely be considered in future work. And another takeaway from this is that for future work, it's important to consider siblings in studies of these owl species, and to ask if this applies to other bird species as well, because again, very little is known about wildlife microbiomes in general. And so being able to characterize these differences and similarities will help with the lack of data on wildlife microbiomes. And it'll also help establish a baseline for the goal of linking wildlife microbiomes with climate change and host health and disease risk. Because we know there are a lot of ties, and some work has been done, but there is a lot more that needs to be done, and we need baseline understanding of variation. So yeah, so there's this idea of the canary in the coal mine. So wildlife microbiomes could be an indicator and actually allow us to understand the effects of climate change on wildlife host species through understanding the effects on the microbiome first. Yeah, so again, we need to understand natural variation in the populations in order to achieve our ultimate goal of understanding, like using communities and populations to understand, sorry, got that backwards, of using individual hosts to understand how climate change is going to impact populations. And yeah, and to be able to potentially predict threats to different species more quickly. So some of our future work is going to be in characterizing wildlife microbiomes in Kenya, community dynamics and migration patterns of birds in Western Asia and the Middle East. This figure is shows the Mediterranean Black Sea flyway, one of the most major migration routes in the world, and zoonotic pathogen pathways and disease dynamics to be tied to this. So the big takeaway for this presentation, hopefully, is that biodiversity is really more than meets the eye. And that the little things in life sometimes matter the most. So the little microbes that are inside us and on our skin, et cetera, are impacting our biology and the biology of everything around us in a huge way. And it's really important to understand those dynamics to be able to conserve wildlife species and also help our own health. And we'd like to thank our co-authors, as well as collaborators and the folks here at the Pajarito Environmental Education Center for inviting us to speak. And we could take any questions now.
Great, thank you so much. So we do have a couple questions. Okay. Let me see if I'm getting all of them. Is there ongoing sampling of the owl species, and will you be keeping track of changes over time?
Yeah, that's a great question. The owl nest boxes are still going. They are run by a collaborator down on the border who just moved down there. So hopefully we'll be monitoring these for a long time. And the ultimate goal of the nest boxes is to get an idea of some of their natural history, life history traits, and that they don't really know about. These are very specifically the whisker screech owl. There's not a lot known about these guys at all. So yeah, I think future work we could consider sampling more. Funding is obviously an issue, and we ran out of it. But we would like to, and we could potentially have them in the freezer and stored. So the great thing about these samples is they can be collected and stored frozen at -80 for a long time, and then come back to in the future when we decide to do something with them or we get some more funding. So yeah, we will definitely be doing that.
Awesome, thank you. Okay, if temperatures were increasing or decreasing over time, how would you expect that to impact bird microbiomes?
Yeah. Oh, go ahead, Kane.
I mean, I would just say, so increasing temperature is one of the most important factors that's involved in climate change on impacting microbial communities, and because they're really sensitive to climate. And the cascading effects of that are pretty significant. So, I mean, I think it's going to impact bird populations in varying ways, right? Because like there's diversity to consider, and then there's also composition. And I think in general, higher diversity is a good thing. And there have been a lot of studies showing that. And increasing temperature makes an environment less hospitable to certain species of microbe. And so that's going to lower diversity, probably. So.
Yeah, and yeah, that's a good, yeah. And then there's this idea of your microbe reservoir in your environment, which is where these things are picking them up, right? And so things like Kane mentioned amphibians and chytrid fungus, and there's some of this reservoir of potential colonizing bacteria that are present in the environment. And if temperatures get too high or something happens, maybe a forest fire, you're going to lose those microbes. They're just going to, maybe they can't stand it, and they can't then colonize amphibian skin and protect them against the fungus. And so there's all these, but in birds particular, I don't think we know a lot about it and what could happen. So this is just one thing, probably a decrease in their microbiome, but how that's going to impact them is yet to be seen. But definitely something to consider and to look into. And that's why we need all this baseline data, is like, things are bad now, but they could be worse in the future. So getting, it's not too late to get baseline data.
Awesome. Someone said they love the title. It's a good title. Okay, are there other teams in the US or other countries who are sampling bird microbiomes?
Yes, there's a lot, actually. Some of them, a lot of the work on microbiomes are chickens. So if you search Google Scholar, microbiomes and birds, a lot of chickens are going to come up, or poultry for consumption and things, and how that's impacting production. There's also experimental studies on birds in terms of disease, virus, like viral shedding and things like that. But in terms of going out and sampling wildlife, I think there's more of that happening, which is great, because we need more data and getting them in databases. But yes, there are people around the world that are starting to get these samples, and maybe there are samples, like I said, in the freezer already. There's a ton of samples in Kenya with our collaborators. They have that go back many years, and now that these technologies exist, we can go back into those samples, do a freezer dive, and potentially get some cool results with that. Yeah, so important to have samples in the freezer.
If anything, we take it. Just kidding. It would, okay, so this is a comment and a question, which I think is actually really interesting. It would be interesting to see if you can apply these techniques to examining soil biomes and how they change with environmental challenges. Have you or others undertaken this?
Yeah, so I know that there is a lot of work going on with that. I think a lot of the work on microbiomes kind of in the environment in general is on soil microbiota. And yeah, because they are definitely like really directly impacted by climate, and they also impact so many different things, like for instance, in plant species that are dependent on fungi, like mycorrhizal fungi, nitrogen fixation. That whole cycle is really dependent on the soil bacteria and other microbes that are present. So kind of that's really, really interesting. And I think there is a lot of work going on that, but there could always be more.
A lot of groups that are studying that at Lano, soil composition and different things like viruses, even soil viruses, and how they're influencing like the nitrogen cycle and things like that.
Very interesting. Are there some microbiota who would actually prefer higher temperatures and potentially outcompete the current organisms if it gets warmer?
Yes, there are, bacteria that do very well. Obviously, there's this ones that are the extremophiles that you read about in like hot springs and things like that, and Yellowstone, and that live in these thermal vents, and
the celebrity bacteria.
Yeah. So those are the ones that are, but they're, even amongst the bacteria, there's variation in all these species, and yes, some will be better able to survive higher temperatures than others. And then those will be able to outcompete. So yeah, it's I think a cool question of, are those ones that will outcompete, are they good? Are they bad? Are they pathogens? And so that's another thing about like rising temperatures, is which ones are going to stick around. And that's we don't know. It's a great question.
It is a great question. Awesome. And it looks like that might be our last question, which is a good one to end on, I think. So I'll just give it 1 second.
That last question, I think that also ties into the difference between like diversity and composition too, and how, like, for instance, the vultures that I talked about, like they actually have really low diversity in their microbiomes, their gut microbiomes. But the specific species of bacteria that they do have make them able to like eat rotting flesh and not get sick. So that's kind of one example of maybe how something like that could positively impact a host species, but I'm sure there are many more where it would be a negative effect too.
Awesome. Thank you for that example. Okay, well, it looks like that is all our questions for the evening. I want to thank both of you for joining us this evening again. Thank you so much for sharing your knowledge about this. It is a very interesting subject to me personally. So it's great. Thank you for coming. And also to all the audience that enjoyed this evening. Thanks for coming.